Hello friends, welcome to SAM Biology class. Sex determination is an important concept for biological science students. Biologically, sex can be defined as the aggregate of morphological, physiological and behavioral qualities that differentiate between male and female. Sex behaves as a Mendelian character. Its inheritance follows the law of segregation. The mechanism by which sex is established is known as sex determination. Sex determination. Various genetically controlled mechanisms that determine sex in different organisms are chromosomal mechanism, male haploidy, genic balance mechanism, single gene effect, and finally cytoplasmic sex determination. First of all, let us discuss about chromosomal theory of sex determination. Chromosomal theory of sex determination. Majority of diploid sexual animals possess a pair of sex chromosomes which are represented by X and Y. X chromosome was first observed by German biologist Henking and was described as X body. X body. Chromosomal theory of sex determination was proposed by Wilson and Stevens. Wilson and Stevens. They named X and Y chromosomes as sex chromosomes, or allosomes, and remaining chromosomes which carry genes for somatic characters were named as autosomes. X chromosome carries female determining gene, whereas Y chromosome has male determining gene. In addition to this, X chromosome also carries some genes for somatic characters. The genes that are located on sex chromosome are known as sex-linked genes, and the characters that they control is referred to as sex-linked characters. Their inheritance is known as sex-linked inheritance that we will discuss later on. One of the genetically controlled sex determination mechanism is chromosomal mechanism, in which sex of the organism is determined by the presence or absence of particular chromosomes. In animal kingdom, several modifications of the chromosomal mechanism are observed. Number 1, sex chromosome undifferentiated. In primitive organisms, sex chromosomes are undifferentiated. In such organisms, sex chromosomes have not been identified, and the genes determining the sex seem to be located on certain autosomes. Second type of chromosomal mechanism is XX female and XY male type. This type of sex determination system is seen in human beings, Drosophila and certain insects. In this system, female individuals possess a pair of X chromosomes and the male individuals possess one X and one Y chromosome. In XX female and XY male system, female individuals have a pair of X chromosomes, so all the gametes formed by the female individuals have the same X chromosome. Because of this, females are known as homogametic. But the male individual possesses two types of gametes. 50% of the gametes possess one X chromosome and remaining 50% possess one Y chromosome. Because of this, male individuals are heterogametic. That type of sex determination system is ZW female and ZZ male type. This type of sex determination system is observed in fowl and other birds, certain fishes, grasshoppers and butterflies. In this sex determination system, female individuals are heterogametic. Female individuals are heterogametic. They possess one Z chromosome and one Y chromosome. But the male individuals are homogametic, produces only one type of gametes, all of which possess one Z chromosome. Because of this, female individuals are heterogametic. They produce two types of gametes. 50% of the gametes possess one Z chromosome and remaining 50% possess one W chromosome. But the male individual produce only one type of gametes, all of which possess one Z chromosome. Fourth type of chromosomal mechanism is XX female and XO male type. This type of sex determination system is observed in squash bug, Onosa. M.C. Klung first observed that male squash bug have 10 pairs of chromosomes. Male squash bugs have 10 pairs of chromosome plus an unpaired X chromosome, whereas the female bugs possess 11 pairs of chromosomes. Because of this, male bugs produce two types of gametes. 50% of the gametes possess only 10 chromosomes and remaining 50% of the male gamete produce 11 chromosomes. But female bugs produce only one type of gamete, all of them possess 11 chromosomes. So when male gametes having 10 chromosomes are fertilized by female gametes, male bugs are formed. But when male gametes having 11 chromosomes are fertilized by female gametes, female bugs are produced. This type of sex determination system is observed in heteropterans and orthopterans. Fifth type of chromosomal mechanism is Habro-Bracon type. Habro-Bracon type. In Habro-Bracon, the sex determination system is similar to wasp and bees. In this case, fertilized eggs develop into females and unfertilized eggs develop into male. In addition, some of the fertilized eggs also develop into males. This is because of the presence of a number of alleles which are XA, XB, XC and XD and so on. So the haploid males which develop from unfertilized eggs could be XA or XB or XC or XD. But the diploid male which develop from fertilized eggs could be homozygous for the alleles. So they could be XA by XA, XB by XB and XC by XC and so on. But the diploid females which are always heterozygous, they could be the diploid females which develop from the fertilized eggs. They are always heterozygous. So they could be XA by XB or XA by XC or XD by XD or XB by XC or XB by XD. So friends, this is about different type of chromosomal mechanism of sex determination. Second type of sex determination mechanism is male haploidy. Male haploidy. This type of sex determination system is observed in honeybees, wasps, ants and certain other hymenopterans where parthenogenesis is widespread. There are three types of individuals: A. Diploid queens. Diploid queens develop from fertilized eggs. They are functional females and they produce ova. And second type of individuals are diploid workers. Diploid workers which also develop from fertilized eggs, but they are underdeveloped non-functional female. They are unable to produce eggs. And third type of individual is haploid drones. Haploid drones or males. Drones are haploid organisms which develop from unfertilized eggs by parthenogenesis. They are functional male. Another type of sex determination mechanism is genic balance mechanism. Genic balance theory was proposed by Bridges, who suggested that all individuals, whether male or female, possess the genes for both maleness and femaleness. But ultimately, which individuals will develop depends on the preponderance of that type of gene. So when male determining genes are more as compared to female determining genes, then male individuals will develop. And when female determining genes are in excess, then the male determining genes, then female individual will develop. In Drosophila, X chromosome carries more genes that incline development toward femaleness, and the autosome carries the genes that incline development toward maleness. So in Drosophila, sex of the individual is determined by the ratio of the number of X chromosomes to the number of autosomes. Another type of sex determination mechanism is single gene effect. Single gene effect is observed in certain organisms such as Neurospora, Chlamydomonas, yeast, asparagus and maize, and certain fishes where a single gene is responsible for the expression of sex. For example, in asparagus, a single gene determines the sex. And here maleness is dominant over femaleness, and males are ordinarily heterozygous. Finally, the last mechanism of sex determination is cytoplasmic sex determination. Biddle and Tatum suggested that in bacteria, sex is determined by the presence or absence of a cytoplasmic factor called F factor. Bacterial cells that possess the F factor or fertility factor are considered as male or donor. They are designated by F+, whereas the bacterial cells that do not have the F factor are considered as female or recipient. They are designated as F-. And during conjugation, a copy of the F factor is transferred from F+ cell to the F- cell. So friends, this is all about different mechanisms of sex determination in different types of organisms. For more updates, subscribe my YouTube channel, Sam Biology Class.